reflex reaction and reflex arc. What is reflex reaction? It is nothing but an automatic, instantaneous, involuntary and unconscious response or reaction to a stimulus. So, why is this response so instantaneous and involuntary? Because it is majorly controlled and coordinated by spinal cord. Majorly controlled and coordinated by, what is this reflex arc? Reflex arc is nothing but a total neuronal components that mediate this reflex reaction. We already learned that reflex reaction is an automatic, instantaneous, involuntary and unconscious response to a stimulus, and this is majorly controlled by spinal cord. Then all the neuronal components that are involved in bringing about this reflex reaction together constitute the reflex arc. Then what is this reflex arc exactly means? It is nothing but a pathway. It is a pathway for the transfer of nerve impulse in bringing about a response, a sudden and instantaneous response to a stimulus. It is a neuronal pathway. The nerve impulse is transferred to the central nervous system CNS, and from there this is reflex arc. Then when you say reflex arc is matter of neuronal components, what are different neuronal components involved in this reflex arc? They are, number one, minimum or at least one afferent neuron, otherwise nothing but sensory neuron. The components of reflex arc are, number one, minimum one if afferent neuron, otherwise a sensory neuron, and number two, one internuncial neuron, and number three, one efferent neuron, otherwise a motor neuron. All these three neurons are arranged in a sequence. Now let us discuss about the transfer of nerve impulse through this reflex arc. The sense organ or a receptor receives external stimulus through this sensory neuron. Now this sensory neuron or efferent neuron convey this impulse received from the sense organs to the central nervous system. Sensory neuron to the central nervous system, that is CNS, that is to the spinal cord. Because we already discussed that simple reflex reactions are coordinated by spinal cord. Okay. Now these impulses, sensory impulses received by the central nervous system are processed, analyzed. Now from the central nervous system, these processed impulses will be sent back to the effectors through the efferent neuron or motor neuron. CNS processes and analyzes received from the receptors or from the receptor. Yes. Now this central nervous system sends motor impulses to the effectors through the motor neuron or efferent neuron. That is how we respond to the stimuli that are received under not conscious control immediately. For example, we can take knee jerk reflex. If something is hit on our knee, our response is so quick, which is the best example for this reflex reaction controlled and coordinated by spinal cord. Knee jerk reflex మనం ఎగ్జాంపుల్ గా తీసుకున్నప్పుడు ఇక్కడ muscle spindle act as a receptor here. This is an internal receptor, and beginning from this muscle spindle we have one efferent neuron going to the central nervous system, and then from the central nervous system, that is from the spinal cord, coming back to the skeletal muscle, we have efferent neuron. The entire impulse, nerve impulse during this reflex reaction, when something is hit on the knee, travels from the muscle spindle to the efferent neuron to the central nervous system, that is spinal cord, and from there the impulse, processed impulse, will be back to the muscle, that is the effector, through the efferent neuron. Here generally efferent neuron enters into the central nervous system, that is the spinal cord, through the dorsal root of the spinal cord, whereas efferent neuron comes out of the spinal cord to the effector through the ventral root of the spinal cord. ఈ టాపిక్లో మనం బయట జరిగే చేంజెస్ బయట ఎన్వైరన్మెంట్లో బయట లేదా శరీరం లోపల జరిగే చేంజెస్ని మన నాడీ వ్యవస్థ ఎలా రిసీవ్ చేసుకుంటుంది, దానితో మనం ఎలా రియాక్ట్ అవుతాము ఈ చేంజెస్కి అనే దాని గురించి నేర్చుకుంటాం. సో లుక్ ఎట్ ద హెడింగ్ సెన్సరీ రిసెప్షన్. హియర్ ద టర్మ్ సెన్సరీ స్టాండ్స్ ఫర్ సెన్స్ ఆర్గన్స్. Reception, receiving. Reception stands for receiving, means the sense organs are specialized in receiving the changes in the environment, whether it could be external environment or internal environment. Okay, then whatever changes in the environment that are received by these sense organs are transferred to or conveyed to central nervous system, where the processing and analysis of this information is done over there, and accordingly the central nervous system sends motor impulses to the effectors. Okay, now you react to that change in your external or internal environment, and your reaction to the changes in the environment, you are calling it as response. Now when you look at all the three components of this heading, reception, processing and response, there are three components. One is receptors, number two it is central nervous system, CNS, and then it is effectors. The changes in the environment received by the receptors are conveyed to the central nervous system through the sensory neurons. This is called as a sensory route otherwise. Now the processed and analyzed information from the central nervous system is conveyed to the effectors through motor neurons. Then when you come to these receptors, there are basically two different types. What are they? They are, number one, external receptors, otherwise called as exteroceptors. External receptors or exteroceptors. Then internal receptors, otherwise interoceptors. Then what is the difference between the two? External receptors are specialized in changes in the external environment, whereas internal receptors or interoceptors are specialized in receiving changes in our internal environment of the body. Now, about exteroceptors. Generally, the sensory perception and processing of the impulses received by external receptors takes place under our conscious control, and it is in turn controlled by somatic nervous system, which you already studied about. So, whatever changes in the external environment that are received by the external receptors or exteroceptors are going to the central nervous system through the sensory neurons, and processed information is sent back to the effectors and you are responding. Now, this sensory perception and response using external receptors is controlled by somatic nervous system that takes place under our conscious control. And next, about interoceptors. Interoceptors are present inside our body. Generally, they are associated with our visceral organs and blood vessels. And whatever impulses received by these interoceptors are also sent to the central nervous system through the same sensory neurons, and response is through the effectors. Right? Now, this sensory perception by the interoceptors and response through its effectors is controlled by autonomous nervous system, which is generally not under our conscious control. We discussed about exteroceptors and interoceptors. Fine. Then what about the effectors? What are the effectors of our body? How are you bringing about the action? Means this is muscles. Effectors of our body are nothing but muscles. Then you said receptors are of two types, external receptors and interoceptors. Then are the effectors for both the receptors are same? Means you get two categories again here. When you say muscles are the effectors, along with the muscles there are also glands. When you come to the muscles, muscles are actually of three different types. They are striated muscles, smooth muscles, then cardiac muscles. Out of these three types of muscles, only striated muscles work under our conscious control, whereas smooth muscles and cardiac muscles do not work under our control. That means the response by striated muscles is voluntary, that is under our control, whereas response by smooth muscles, cardiac muscles and glands, that means your response is not under your conscious control. They are number one, exteroceptors, as we already discussed. Number two, interoceptors or internal receptors. Number three, proprioceptors, exteroceptors, interoceptors, proprioceptors and thermoceptors or thermoreceptors. But exteroceptors, why do you call them as so? Because they lie very close to our body surface, means they are on the body surface or close to the body surface, so they are specialized in receiving changes in the external environment. They lie or present on the body surface or close to the surface of the body. We take the examples: eyes, nose, ears, tongue and skin comes under exteroceptors. You already studied them as 5 special sense organs. That means they are specialized in the sight, hearing, smell, taste, touch. Skin is for touch, pain, pressure perception, etc. Next, interoceptors, present inside our body as a part of our visceral organs and also associated with the blood vessels. present in the visceral organs. Generally, these internal receptors or interoceptors are chemoreceptors, specialized in receiving changes in the chemical nature of the substances. Next, it is proprioceptors. These proprioceptors are also a type of interoceptors. Also interoceptors basically. Then where are they present and what for are they? These proprioceptors are generally present in the muscles, in the tendons, in the joints as well as in the internal ear. Location of proprioceptors, muscles, okay, that is location. Then what for are they? They are specialized in perceiving body orientations and movement. Sensitive to, that's about proprioceptors. Then thermoceptors or thermoreceptors. They are specialized in sensitizing or sensing changes in the temperature. There are two types. One is caloriceptors. Thermoreceptors are of two different types. They are, number one, caloriceptors. Caloriceptors are specialized in receiving heat, and frigidoceptors are sensitive to cold. Human eye is the organ of vision. The eyes are lodged in specialized sockets in the facial region of the skull called as orbits. The structure of the eye can be studied under two heads. One is accessory structures and number two it is eyeball. Lodged, eyes are lodged in specialized sockets of the facial skull bones called as. What are the structural details of the eye? Let us study the structure of eye under two heads. One is accessory structures and number two, eyeball. What are the accessory structures associated with the eyeball? Let us learn these accessory structures under different heads. Number one, it is eyelids, eyelashes, and eyebrows. Number two, it is lacrimal apparatus, as I directly say, lacrimal glands associated with the eyeball. Now, the secretions of these lacrimal glands, you call it as tears. When we discuss about the composition of the tears, it is chiefly made of water, salts, and one bactericidal enzyme, that is lysozyme, an enzyme specialized in killing the bacteria that enter our eyes. Next, associated with the eyeball, there are also muscles. Muscles present outside the eyeball and connecting the eyeball with the orbits, you call them as extrinsic muscles. Extrinsic muscles. These extrinsic muscles are basically of two different types. They are, number one, oblique muscles, and number two, rectus muscles. Oblique muscles and rectus muscles are the two types of extrinsic muscles that connect the eyeball with the orbits and play a role in the movement of the eyeball. Now, these muscles, oblique and rectus muscles, are again of different types based on their position and orientation. When you take oblique muscles, there are two types. Number one, superior rectus muscle oblique muscles, and number 2 it is inferior. Superior and inferior oblique muscles. Then rectus muscles are of 4 different types. They are superior, number 2 inferior, number 3 medial, and number 4 it is lateral rectus muscles. Now these muscles, extrinsic muscles, both oblique as well as rectus muscles, are supplied by the cranial nerves. Then out of this superior and inferior oblique muscles are supplied by the third pair of cranial nerves. The same way superior, inferior and medial rectus muscles are also supplied by the same third pair of cranial nerves. Then superior oblique muscles are supplied by the fourth pair of cranial nerves, whereas lateral rectus muscles are supplied by sixth pair of cranial nerves. You know, third pair of cranial nerves are nothing but oculomotor nerves. Then fourth pair are trochlear or pathetic nerves, whereas the sixth pair of nerves are nothing but abducens nerves. Then when we come to the structure of the eyeball, the wall of the eyeball is basically made of 3 layers. The wall of the eyeball has 3 layers. The 3 layers from outside to inside are outer fibrous tunic, middle vascular tunic. The outer layer is fibrous tunic, middle layer is vascular tunic and inner layer is nervous tunic. This vascular tunic is otherwise called as avia and nervous tunic is otherwise called as retina. Now let us learn in detail about all the 3 layers of the eyeball as well as a lens present inside the eyeball. Out of the 3 layers of the wall of the eyeball, the outermost layer is nothing but fibrous tunic. Outer layer is fibrous tunic. Now this fibrous tunic has 2 regions. Tunic stands for a layer. This outer layer, fibrous tunic, has 2 parts. Anterior cornea and posterior sclera, otherwise called as white of the eye. Now about cornea. This is a non-vascular, the anterior part of the fibrous tunic. This is a transparent layer. Though not a lens, it acts as a fixed lens, allow the light to pass through or to enter into the eyeball. Now this cornea is externally covered by a thin membrane, you call it as conjunctiva. Cornea is externally covered by a thin membrane called as conjunctiva. Internally, the cornea itself covers the pigmented part of the eyeball, that is nothing but iris. So it covers iris, the pigmented portion, and it is covered by a thin membrane called as conjunctiva. Next, it is about posterior sclera or white of the eye. This sclera covers most of the part of the eyeball and it is very tough. A tough layer gives strength to the eyeball. This same sclera protects the internal parts of the eyeball, and it also plays a role in maintaining the shape of the eyeball. So, this is about the outer fibrous tunic with anterior cornea and posterior sclera. Now, at the junction between the two, at the junction between outer or anterior cornea and posterior sclera, you find one canal here. This is called as canal of Schlemm. Followed by this outer fibrous tunic, it is middle layer vascular tunic, or otherwise called as uvea, uvea or avea. This middle layer vascular tunic in turn has three parts. What are those three parts? Out of the three parts of vascular tunic, the posterior one is choroid. The posterior most part of middle vascular tunic is nothing but choroid. This is a pigmented layer, vascular layer, has a rich supply of blood, then it is somewhat bluish in color. If this is the choroid, anteriorly this choroid has a thickened portion, you call it as ciliary body. Now, this ciliary body has one ciliary process that consists of muscles, a ring of smooth muscles called as ciliary muscles. It has one ciliary process. Now, this ciliary body, which is also pigmented, consists of a ring of smooth muscles called as ciliary muscles. Now, this ciliary body and the ciliary process play a key role in holding the lens with the help of a ligament. You call it as suspensory ligament because it is useful to suspend or hold the lens in position. Ring of smooth muscles called as ciliary muscles. Then, what is the significance of these ciliary muscles? We already learnt that ciliary process is holding the lens with the help of one suspensory ligament. And what for else? These ciliary muscles play a key role in eye accommodation power. Play a key role in eye accommodation power. What is this eye accommodation power? It is nothing but changing the focal length of the lens with the changing distance of the object. Eye accommodation power is nothing but a power or ability to change the focal length of the lens with the changing distance of the object. Suppose the object is very close to our body, then ciliary muscles contract, so the lens become more convex. Lens is already a convex transparent structure, this become more convex. When the distance of the object increases, the convexity of the lens decreases. So, eye accommodation power is facilitated by these ciliary muscles. Next, and the third part of this vascular tunic is nothing but iris, the pigmented portion of the eye. Okay, and where is this iris present? The iris is present suspended, otherwise you say, between the cornea and the lens. It is present between the cornea and lens and is attached to the ciliary process. Now, this pigmented iris has its own muscles called as iris muscles. Then, at the center of this iris, you can find one space or a hole. You call this hole present at the center of the iris as pupil. Now, what is the significance of these iris muscles? Means The iris muscles play a key role in changing the diameter of this pupil, nothing but the hole present at the center of the iris. Thereby, they control the amount of the light that falls on the retina. That is, the amount of light that passes through the lens, vitreous chamber and on the retina. So far we discussed about the two layers of the wall of the eyeball, that is inner fibrous tunic and middle vascular tunic. Now let us learn about the innermost layer, that is nervous tunic or retina. This inner layer or retina in turn has two portions. What are those two? An outer layer close to the choroid is called as non-nervous portion, or you can call it as non-visual portion. Why are you talking about this non-visual region? Because you know retina is nothing but area of formation of images. But out of the two layers of retina, the outer layer is non-nervous portion and here no images will be formed. That is why we are calling it as non-visual portion. Then inner one is nervous portion. Now when you say this outer layer or outer region is non-visual or non-nervous, then what is it made of and what for is it? Actually this is made of one pigmented epithelium. Made of pigmented epithelium. When you say okay epithelium, fine, we know. Then what pigment do you find here? Means the pigment is melanin. And what is the significance of this melanin? What for is this melanin? Means it absorbs the light and prevents the penetration of the light from the retina to the outer layers of the eyeball. And next about inner nervous portion or visual portion of the retina. Now this visual portion of the retina is made of two different types of photoreceptor cells. What are those photoreceptor cells and what is their role in the formation of images? The two types of photoreceptor cells are number one rods and number two it is cones. Then about the rods, as we already discussed that these are photoreceptor cells specialized in receiving the light and formation of images. Now about rods, these rods are comparatively more in number than that of the cones. Now they consist of a visual pigment called as rhodopsin. Otherwise called as visual purple. Now this rhodopsin present in the rods is specialized in dim light vision, otherwise you call it as twilight vision, otherwise scotopic vision. Rods with the pigment rhodopsin or visual purple are specialized in dim light vision, otherwise called as scotopic vision, otherwise called as twilight vision. About this pigment rhodopsin, this rhodopsin is actually made of a protein opsin in combination with one retinal. Opsin is the protein part and retinal is nothing but derivative of vitamin A. Now this is a reversible product. Cones consist of another pigment, visual pigment called as iodopsin, otherwise called as photopsin. Now these cones are specialized in bright light vision as well colored vision. When rods are specialized in dim light vision or scotopic vision, cones are specialized in bright light vision as well as colored vision. And the other thing is, unlike the rods where they are all alike, cones are of 3 different classes, 3 different types. And these 3 different types of cones are differentially sensitive to 3 different colors, that is red, green and blue colors. Sensitive to, differentially sensitive to red colors, red, green and blue. That means, can we see only these 3 colors? No, but we can see all other colors, right? So, formation of images of all other colors is nothing but a combination of these 3, just like prime colors. Next, suppose when all the 3, we said they are differentially sensitive to 3 different colors. Suppose they are equally sensitized. When all the 3 classes of cones are equally sensitized, then the image formed is white in color. This is called as a trichomacy concept. On equal sensitization of all the 3 types of cones, image formed is white in color. The nervous portion or visual portion of the retina. This visual portion of the retina in fact has 3 layers. The 3 layers are outer photoreceptor layer, middle bipolar cell layer and inner one is ganglion cell layer. Whatever rods and cones, that is photoreceptor cells of visual portion we discussed, come under outer photoreceptor layer. About the retina once again. So when you look at this retina, towards the posterior part of the central region of retina, you find a region called as macula lutea, otherwise called as yellow spot. The posterior central part of the retina, you call it as macula lutea or otherwise yellow spot. Now, at the center of this yellow spot, you find one shallow depression called as fovea centralis. Then, what is the speciality of this fovea centralis? When we discussed about photoreceptor layer of the visual portion, we said there are both rods and cones. Rods are specialized in dim light vision and cones are specialized in bright light and colored vision. But when you come to this fovea centralis, it has only cones. It has only cones, no rods. That means it is for bright, sharp vision. Vision required during reading, driving, etc. Present in the cavity of the eyeball, held in position or suspended by the ciliary process. There is a transparent and biconvex non-vascular structure, nothing but the lens. Now, this lens divides the cavity of the eyeball into 2 chambers, where an anterior chamber is called as aqueous chamber and the posterior chamber is called as vitreous chamber. So, the lens divides the cavity of the eyeball into anterior aqueous chamber and posterior vitreous chamber. Both the chambers are filled with the fluids. The fluid present in the aqueous chamber is somewhat liquid, watery, where you call it as aqueous humor, whereas the one present in the vitreous chamber is made of jelly type of tissue, where you call it as vitreous humor or vitreous body. Next, about aqueous humor. This aqueous humor is actually secreted by ciliary process, the process of ciliary body, and is useful to nourish the cornea and the lens. Secreted by ciliary process and is useful to nourish the cornea and the lens. Then about vitreous humor, generally made of jelly-like connective tissue. Now, this vitreous humor along with aqueous humor plays a key role in maintaining the shape of the eyeball. How? This vitreous humor present in the vitreous chamber applies pressure on the wall of the eyeball from within, where you call it as intraocular pressure. Now, this intraocular pressure posed by vitreous humor along with aqueous humor on one hand, and the outermost layer of the wall of the eyeball, that is sclera or white of the fibrous tunic, which is giving support to the or toughness to the eyeball. Both of them play a key role in maintaining the shape of the eyeball. Okay. So, on one hand, vitreous humor is providing pressure on the wall of the eyeball, you call it as intraocular pressure. On the other hand, the sclera of the eyeball, being very tough, made of fibrous tissue, is giving toughness to the eyeball. So, both of them together play a role in maintaining the shape of the eyeball. This is about the structure of the eye. Then it is about formation of image. So, we finished learning about the structure of the eye. Now, let us learn about working of eye. When the light falls on the cornea, this light passes through the aqueous humor, then it is pupil of the eye, nothing but the hole present at the center of the iris. Then it is lens. Through the lens it enters vitreous chamber, where from it falls on the retina of the eye. As we already discussed about, the visual portion of the retina has 3 layers. What are those 3 layers? The outer layer is nothing but photoreceptor layer, and the middle one is bipolar cell layer, and the inner one is ganglionic cell layer. That means, on which layer of the retina light falls first, means it falls on ganglionic layer, then it passes through bipolar layer, then it reaches the photoreceptor layer. Then we already discussed that photoreceptor layer has rods and cones. Both rods and cones have their respective visual pigments called as rhodopsin and iodopsin. Now, when the light falls on these rods or cones, their visual pigments gets dissociated. For example, let us say it is rhodopsin. Rhodopsin is a reversible product we already discussed. When light falls on these rods, rhodopsin get dissociated into opsin plus retinal. Now, this dissociation of retinal from opsin causes depolarization of the cell membrane of the rods. Causes depolarization of the cell membrane of rods. Now, this depolarization lead to generation of action potential. Now, this action potential, which is developed in the photoreceptor layer, will come back to bipolar layer, then from there to ganglionic cell layer. Now, the ganglionic cell layer, we know that it is the innermost layer. Now, the axons of all the axons of this ganglionic layer join together to form one optic nerve. Now, this optic nerve comes out of the eye, and this optic nerve will be supplied to one visual cortex present in the occipital lobe of cerebral hemispheres, where the impulses, sensory impulses, that is optic impulses received are analyzed, processed, and accordingly the image is formed. Now, about this movement or this optic nerve coming out of the eyeball. The point of the eyeball where this optic nerve comes out, you don't find retina. Retina is absent. That means no images are formed at the point of origin of optic nerve from the eyeball, where this is called as blind spot, otherwise called as optic. So, this is about how the images are formed. Then, let us just discuss about the sequence of layers of wall of the eyeball. So, when you start with retina, the innermost layer, retina has how many layers? When you consider retina or nervous tunic as innermost layer, it has how many layers? We discussed about the inner nervous tunic has 3 layers. What are these 3? Number 1, ganglionic cell layer facing the vitreous chamber, then it is bipolar cell layer, then it is photoreceptor layer. So, all the 3 comes under nervous portion. Then followed by this non-nervous portion, that is pigmented epithelium containing melanin. Then this is choroid. Choroid is followed by the sclera. So, this is the sequence of layers in the wall of the eyeball. So, from inside to outside, ganglionic cell layer, bipolar layer, photoreceptor layer, followed by non-visual portion of the retina. Then it is choroid and sclera. This is sequence question. We learnt about reflex action and reflex arc, sensory perception, processing and response, and also structure and working of eye.